Hello dear students, welcome back to Sajjan biology classes. In today's session, we are going to discuss about the cloning vectors. And first of all, what are vectors? And vectors, something which they carry are called on vectors. Here, the plasmids, we are considering them as vectors because they are carrying that alien piece of DNA which we have inserted in that. In generally, the bacteriophages or the plasmids, they undergo the multiplication in a bacteria in large number by controlling the nucleic acid material called DNA. And one of the important thing is that the plasmid number, it may vary from one species to other species. You can see here that we have taken an E. coli bacteria diagram here, and where that the bacteria which is consisting of the nucleic acid material called an DNA, and along with that DNA we can see the number of plasmids. Okay, and these plasmids, so one of the plasmid I magnified here, so and this is called an PBr322, plasmid of Bolivar and Rodriguez. So this plasmid we are extracting from the E. coli. So when you look into the plasmid of this diagram, so you can see that PST1, PVU1, EchoR1, CLA1, HIN3, BAMH1, SAL1 and PVU2. And what are all these things? So these are nothing but what we used to call as a cloning sites. So when you look into the plasmid diagram, so these are the following features we are going to study. The first one is that origin of replication, and second one is that selectable markers, and third one is that cloning sites, and fourth one is called vectors for cloning genes in plants and animals. And the first thing we must try to understand here that the plasmid which is having 1, 2, 3, 4, 5, 6, 7, 8 cloning sites, and these are the sites where restriction endonucleases can perform or cuts the specific sites. Okay, these are what that the cloning sites we are calling them. Okay, and second one is that the plasmid which is consisting of two selectable markers. So that one is that ampicillin resistant gene, and second one is the tetracycline resistant gene, and these two act as selectable markers. We discuss later about all these things. And third one is that origin of replication. As we already know that the origin of replication, it won't take place wherever actually it want. It takes place specifically where that the replication fork, what we used to call, so means the replication sites. So in that reason only that origin of replication it begins. And fourth one is that ROP, the replication of proteins. Actually it helps in the replication of plasmids. So let us discuss in detail about the features. First one is that origin of replication. What is this origin of replication? Any alien piece of DNA when you link to this plasmid, so this origin of replication, what it will do, it starts to produce the replication of the alien piece of DNA in the host cell. And this origin of replication site, it also controls the copy number of the linked DNA. Actually each plasmid, so it is having a specific production or specific number of copies it produces. So one who wants more number of copies, they can choose the particular vector or the plasmid which supports actually the high copy of number, and they can select the plasmid. Okay, and next one is that selectable marker, and these selectable markers are essentially helps to identify and to eliminate the non-transformants, and where actually these two allows the permission to the growth of transformants. First of all, what are the transformation? So transformation is nothing but what? Any alien piece of DNA which is transformed into host cell, it is called a transformation. And these two tetracycline and ampicillin resistant gene, they helps to identify and eliminate the non-transformant. Which one is the transformant? Which one is the non-transformant? It is very simple. If alien piece of DNA, if we link to the plasmid, so it becomes a transformant. Only a plasmid, if it is there, it is a non-transformant. That's it. Okay. And next we are going to discuss about the cloning sites. And one more thing, and these genes, they codes for what? Specifically the antibiotic resistant gene. They codes for antibiotic resistance. What kind of antibiotic resistant genes? Genes. The ampicillin resistant gene and tetracycline resistant gene, both they codes for antibiotics like kanamycin, ampicillin, tetracycline, like that. And let us discuss next one is that the cloning sites. So we have totally that 8 cloning sites. Among these 8 cloning sites, only one cloning sites we required to avoid to producing the multiple copy of the fragments. Okay, and we have selected here that BAM-H1. If you insert the alien piece of DNA at the BAM-H1, what it happens, the tetracycline resistant gene is lost. So what it happens if it lost a tetracycline resistant gene? And it is not so easiest job to identify whether the plasmid is the non-transformant or transformant. To identify the transformant and non-transformant, we used to do one small procedure in a laboratory. So let us discuss here. In the laboratory, they have conducted one small experiment. So what I have done, so the one petri dish which is containing the media and ampicillin, and another petri dish which is containing the media along with that the tetracycline. The transformants which are transferred here. So because we need to identify which one, whether the plasmids are transformant or non-transformant, isn't it? So here the transformants, if we allow to grow in a media containing ampicillin, they grow because they have an ampicillin resistant gene. But the transformant, when we transfer to the tetracycline media, so they won't grow because they have lost the tetracycline resistant gene, hence they do not grow in the media which is containing tetracycline antibiotic. Okay, and in this way actually the transformants and non-transformants actually we used to identify. So this is the very cumbersome and very tedious procedure to do whether that plasmid is a transformant or non-transformants. To avoid the complex procedure to identify whether the recombinants or non-recombinants, they have developed selectable markers, and these selectable markers helps to identify the recombinants from the non-recombinant ones in the presence of chromogenic substrate by producing its color. So already here the plasmids, they are provided with the beta-galactosidase enzyme in the resistant gene or the cloning sites. And when the alien piece of DNA is inserted, that the beta-galactoside is become inactivated. So this is called an insertional inactivation. So due to the insertion of alien piece of DNA, that beta-galactosidase, it becomes inactivated, and this is called an insertional inactivation. Once after the formation, means when the beta-galactoside it become inactivated, it produces the blue colored colonies. If that plasmid does not have any kind of insert, it do not produces any kind of the colonies. I hope you understood. And next we are going to discuss about, so what is this? The cloning of genes in plants and animals. And here that cloning of genes in plants and animals, we have taken the 2 examples. One is that the Agrobacterium tumefacium. And Agrobacterium tumefacium, what it do? So let us discuss here. So here that Agrobacterium tumefacium bacteria, which is having a tie plasmid. So the tie is nothing but tumor inducing plasmids, and these tumor inducing plasmids itself can convert the normal plant cells into tumor cells. They have a capability. So this is the thing which was naturally happening in the plants. But and this method is used by the genetic engineering technology, means they have manipulated to produce the desirable gene in the plants. And the best example in the, as we have studied already in the RNA interference, how actually these Agrobacterium tumefacium plasmids, they provide a protection to the plants. So we have manipulated the plasmids in order to provide a protection to the plants. Like that, here that the many of the animals, especially that retroviruses, what we used to call, and these retroviruses, they also used to transfer the specific gene. Okay, and for that we have studied the gene therapy. So where in the gene therapy, what we used to do that by using the retroviral method, we are transferring the specific gene. So like that, so cloning of the genes in plants and animals also provided a great knowledge in the case of human welfare to produce a desirable gene in multiple copy of the number. And this is what about the cloning vectors. And still, do you have any doubts regarding this? So please drop your doubts and comments in the comment section. Definitely I will reply for your queries. Thank you, and please subscribe, like and share. Thank you.